Welcome to a webinar organized by the EU-funded IMPRESS project, focusing on finding ways to produce sustainable chemicals and materials. Our webinar is titled New High-Value Plant-Based Products from IMPRESS. My name is Stanislava Petrova, and I will lead you through it today. The IMPRESS project was launched in September 2019, and it aims to create a new hybrid biorefinery process. It means researching and combining selected key technologies and using them to refine renewable resources such as forestry and agricultural residues into multiple sustainable chemicals and materials that can replace fossil-based products. The IMPRESS project consortium consists of 10 outstanding partners around Europe and is led by Avantium in the Netherlands. Our first webinar, we introduced the IMPRESS consortium concept and the bigger chain reaction that it is part of. If you missed the first webinar, you can find the recording on our homepages. This is the second webinar and it's a series of 4. In this webinar, you will hear the insights on the technologies applied in the IMPRESS project and the interest of using the selected separation and purification processes in 3 advanced value chains of lignin, hemicellulose, and glycols. The first value chain, value for lignin, will be presented by Monika Österberg from Aalto University. The second value chain, recovery of Xylitol, includes multiple partners and is presented by Yannick, Krauke, and Svea, Stephan from KNAUER. And in the third value chain, glycos to valuable end products, you will hear from Siu-Ha Tang from Avantium. However, before diving into the value chains, first in our agenda is the efficient upstream processes. We begin with a video that will introduce us with the primary process of the IMPRESS concept, the Avantium's down technology, the foundation for the described value chains. The video will follow a presentation from Niels van Linden from Lenntech, who will introduce us with the treatment of sugar solutions by membrane-based technologies.
The Dawn technology process. The Dawn technology process lets us convert wood chips into a variety of valuable products, including sugars for industrial use. How, you're thinking? Well, we've found out that we can extract high-quality glucose, wood extractives, and clean dry lignin from wood chips using a concentrated acid low-temperature method. The core of the Dawn technology process is the wood hydrolysis section. This takes place in 2 stages, the pre-hydrolysis and the main hydrolysis. In the pre-hydrolysis, we start by filling a reactor with dry wood chips, then flooding it with a mixed sugar and acid stream from a previous reactor. We then take off some mixed sugars for further processing. We replace the volume of mixed sugar solution taken off by pushing in fresh 37% HCl and push out the remaining HCl with a displacement fluid. Now that we have extracted the mixed sugar stream, let's take a look at the main hydrolysis. In the main hydrolysis, we convert wood cellulose into a glucose stream which flows out of the reactor. All that remains is a clean lignin product. We pick up from the point where the displacement fluid was last used to push out the mixed sugars. We use a glucose and acid stream from a previous reactor to push out the displacement fluid. We then collect a glucose product stream from the reactor. We replace the volume of glucose product stream that was taken off by pushing in fresh 42% HCl. Finally, we push out all of the glucose product stream with displacement fluid, leaving behind clean lignin, which is then discharged from the reactor, ready for it to be filled with clean wood chips to restart the cycle. This last step completes the process. As you can see, the Dawn technology process is a series of operational steps in which various phases cycle around the reactor chain. This kind of operation is known as a simulated moving bed, where we use countercurrent flow of the HCl and other process streams to effectively fractionate the woody biomass into mixed sugars, lignin, and a high-purity glucose.
Welcome all to this part of the Impress webinar on the treatment of sugar solutions by membrane-based technologies. I am presenting this part of the webinar on behalf of Lenntech Water Treatment Solutions. As you have seen in the previous video, in the Dawn process conducted by Avantium, various sugar solutions are produced. These sugar solutions have different compositions based on the one hand, the feedstock, so the original materials that are fed to the hydrolysis process, and secondly, to the operation and the conditions in the pre-treatment, such as the temperature and the amount of acid that is used to hydrolyze the feedstock. In the Impress project, different sugar solutions are produced, of which the first sugar solution is called the C6 solution, which contains mostly glucose, and secondly, the C5, C6 sugar solution, which contains mostly xylose and glucose sugars. The sugar concentration in these solutions typically range between 10 and 40 grams per liter, whereas there's also ions in these sugar solutions, and the concentration of the ions is approximately 10 to 20 grams per liter. Finally, the color of these sugar solutions ranges from black to brown or even a bit yellow or colorless. As you can see on the top right, at Lenntech, we have been treating sugar solutions with different colors, all depending on whatever pre-treatment is applied and what the hydrolysis conditions were. On the left, you can see a very black solution, and on the right, you can see an almost colorless sugar solution. The objective of treating these sugar solutions is to make the sugar solutions available for the downstream processing in which xylitol and glycols are produced. In order to make these sugar solutions available for the production of these products, on the one hand, the ions must be removed. We are doing tests at Lenntech with different technologies, such as electrodialysis technology, nanofiltration, and membrane distillation. All these three technologies are membrane-based technologies, of which we will now explain more. If we are speaking about electrodialysis, we have a working principle that is driven by an electric potential. So applying electricity and using a combination of ion-permeable membranes, so-called cation exchange membranes and anion exchange membranes, we can have the feed solution, which contains the sugars and the ions in the blue channels, and by applying an electric current, the ions are transported from the feed solution to the so-called concentrate solution, which initially is water. As you can see in the schematic here on the right, the ions are transported from the feed solution to the concentrate solution, leading to a diluted solution, which is called the diluate, which contains still the sugars, but from which the ions are removed. In addition, you have the water solution, which starts as water and then ends up as a solution where the ions are collected and is called the concentrate. The idea behind this is that the ions are mobile under an electric field, but that the sugars, here indicated in the green circles, are not mobile or are not affected by the electric potential and therefore remain in the feed solution. When we are doing tests at Lenntech to optimize and test this technology, you can see here an example of an experimental setup. In the center, you can see the membranes. And on the top, you can see here the feed solution. In this case, this was a brown sugar solution, and here a transparent solution where we are going to collect all the ions. Here you can see a graph of a typical experiment. On the horizontal axis, you can see the runtime of the experiment, And on the vertical axis, you can see the ion concentration in grams per liter. You can see that from the diluate, which starts as the sugar solution with the ions are removed as a function of time. And that in the concentrate, the ions are collected, and therefore the ion concentration in the concentrate increases over time. So what are the key performance indicators for this technology? At Lenntech, we can allow for more than 95% of ion removal, while we are only losing less than 2% of the sugars from the feed solution to the so-called concentrate. When we are subsequently are going to concentrate the sugars with, for example, nanofiltration, we are applying pressure in a setup where we have a different membrane as compared to the electrodialysis membranes. We here have a water permeable membrane. So by applying pressure, we can push the water through the membrane while we are rejecting the sugars. Again, we are working with an initial incoming feed solution, which contains the sugars. We produce a so-called permeate, which is the water that permeates through the membrane. And we obtain a concentrate, which is a residual water stream in which the concentrated sugars are collected. When we are doing tests at Lenntech, you can see here again an example of an experimental setup. With here in the red circle, you can see the actual membrane modules in which the membranes are placed. When we are doing tests, we are looking for this technology at the water removal or the water recovery percentage. The water recovery percentage relates the volume of the collected permeate, so the clean water, with respect to the initial volume of the feed solution, the diluted sugar solution. On the right here, you can see again a graph with on the horizontal axis the water recovery percentage. So this progresses throughout the experiment. We are pushing more and more clean water through the membrane, and therefore the water recovery percentage increases. Because we are pushing more water out of the feed solution and we are rejecting the sugars, you can see that as a function of the increasing water recovery, the sugar concentration factor is starting to increase. We are therefore concentrating the sugars. For some tests, we achieved more than 87% water recovery, which relates also to a sugar concentration factor of around 7 times. Because the membranes are very tight, we can keep the sugars in the feed solution, and therefore we can limit the sugar loss to only 2%. In another technology, we are using membrane distillation. Membrane distillation is driven by heat, and these membranes are vapor permeable. So these membranes look very similar to nanofiltration. However, instead of water that can permeate as a liquid, we can here have water permeating as a vapor. So these are therefore also different types of membranes. Sugars cannot be volatilized, so they cannot become a gas, and therefore they are rejected by the membrane. On the other hand, water can be volatilized or vaporized, and therefore if you have a high vapor pressure at the feed side of the membrane and a low vapor pressure on the permeate side of the membrane, we can remove water from the feed solution. Again, the feed solution is the sugar solution, and in the permeate we obtain actually a vapor, and on the concentrate side we can again collect the concentrated sugars. Again, if we are doing tests at Lenntech, you can see here an example of the experimental setup where we have here indicated the membrane module. Again, we can do a very similar experiment as for nanofiltration, where on the horizontal axis, you can see in this graph the water recovery, whereas on the vertical axis, we can again see the sugar concentration factor. By increasing the water recovery, in other words, by removing more and more water, but this time as a vapor, we can again concentrate the sugar in the feed solution. We achieved 86% of water recovery, which corresponded in this case to approximately 8 times concentration factor. And again, we could keep the sugars within the feed solution, limiting the losses of sugar again to 2%. So what are the takeaway messages from this part of the webinar? Membrane-based water treatment technologies can effectively use to make the hydrolyzed sugar solutions produced by Avantium in their Dawn technology suitable for the downstream processing that will be conducted by other partners in this consortium. We can achieve 95%, more than 95% iron removal by electrodialysis while only losing 2% of the sugars, and in subsequent nanofiltration or membrane distillation technology, we can concentrate the sugars by approximately 8 times, while again limiting the losses of sugar to 2%. Currently at Lenntech, we are scaling up our technologies from lab scale units to pilot scale units, and we are also working on the designs to further bring this technology to practice. Thank you for your attention and good luck with the other webinars.
Thank you, Niels. Next, we shall hear 3 interesting presentations of the Impress concept value chains. Our next presentation deals with the lignin stream and an example of what value products it can offer. Monika, please.
My name is Monika Österberg. I'm a professor in bioproducts chemistry and head of the Bioproducts and Biosystems department here at Aalto University. So in light of the recent increasing consumption of raw materials, there is an urgent need to find more sustainable solutions from renewable materials. And here at Aalto, we are looking at different ways of developing new bio-based materials. And here we see lignin as a great solution. Lignin is a polyphenolic biopolymer found in woody parts of trees and other vascular plants like straw. Lignin makes up 10 to 30% of the lignocellulosic biomass, but it's still mainly treated as a side product from pulp and biorefinery. It's used for low-value applications such as for heat and electricity. However, this is changing, and an interest in high-value applications of lignin is increasing. In this Impress project, non-food agricultural and forest residual biomass is converted into industrial sugars and lignin by Avantium using Dawn technology, which is based on acid hydrolysis. After all the sugars have been taken out of the process, high amount of residual lignin can be produced, typically 20 to 25% depending on the wood species. The aim of the Aalto University Bioproduct Chemistry Group is to study the application of this lignin raw material for high-value materials. For optimal performance in applications, the lignin should ideally be easy to handle, reproducible, well-characterized, and easy to modify. At Aalto, we first analyzed the lignin batches produced by Avantium by using spectroscopic methods, for example NMR spectroscopy and gel permeation chromatography. Here, the samples is placed in high magnetic field NMR, and by this way information can be obtained of the macromolecular structure of lignin polymer, such as interconnecting bonds and the amount of reactive hydroxyl groups. These techniques give us a deeper understanding of lignin structural properties and its reactivity for further modification and application possibilities. So the challenge of lignin valorization have so far been that it has such heterogeneous structure depending on the biological source or also the process from where we are getting the lignin. So lignin is always a side stream, so the processes have been optimized for the cellulosic part, to get, for example, strong and flexible cellulose fibers for pulping and for paper making, or to get the highest yield of carbohydrates in bioethanol production, for example. We here at Aalto work at overcoming these challenges, so we want to take these industrial side streams and develop spherical lignin nanoparticles that are easy to use both in high value applications like biomedical applications, and in high volume applications like adhesives or composites. The hardwood biorefinery lignin received from Avantium is first purified to remove the processing residues and then dissolved into the binary solvent. This binary solvent consists of an organic solvent and a small amount of water. The lignin solution is then added into water, acting as an antisolvent, and on contact with water, lignin spontaneously assembles into spherical nanoparticles. The largest and most hydrophobic lignin molecules form the core of the particles, and the smaller and more hydrophilic molecules are enriched at the surface.
Hello, my name is Maria Moritz. I'm a postdoctoral researcher in Monika Österberg group, Aalto University, and I would like to present you characterization methods of lignin nanoparticles. Lignin nanoparticles are made by solvent shifting, possess a spherical shape, high surface area, and well-defined surface structure. The first characterization which we do is a measurement of size of these particles. For this purpose, we use dynamic light scattering (DLS) technique. This method provides us with the average size of particles, size distribution, but it doesn't give us information about morphology of the particles. Also, we can suspect that we have aggregates in our sample if we see some unexpected results, unexpected measurements. For morphology of the particles, we use imaging techniques such as electron microscopy and atomic force microscopy. For example, scanning electron microscopy provides us overview of morphology of the sample. And transmission electron microscopy, besides morphology, allows us to look at internal structure of the sample. As for AFM, AFM provides us morphology, dimensions, 3D structure, and also we can say something about mechanical properties. Working principle of AFM is scanning of the surface with a scanning probe, which consists of cantilever and sharp tip. Sharp tip interacts with the surface and deflects a cantilever. Deflection of cantilever is recorded by the laser, which reflects from the cantilever and hits the photodetector. This data is used for the topography imaging. In our work, we usually use AFM in tapping mode. The zeta potential is an important indicator of the dispersion stability. This surface potential is measured by modification of DLS. Lignin nanoparticles have a negative surface charge, and repulsion between particles make the dispersion stable. Lignin nanoparticles are easy to handle, and its surface are suitable for modifications. Some molecules, for example chitosan or some proteins, can be adsorbed to the negatively charged surface of lignin nanoparticles. The spherical lignin nanoparticles overcome many of challenges of original lignin and even open up new application possibilities.
Here at Aalto, we are using these spherical lignin nanoparticles for various applications. For example, emulsion stabilizer, as adhesives or wood coatings. and also in more unique applications like hydrogel stabilizing for biomedical applications or stabilizing phase-changing materials for energy storage.
Thank you, Monika. Our second value chain is xylitol recovery from the hemicellulose stream. Yannick and Svea, please.
Hello, my name is Yannick Krauke. I'm an application specialist from the KNAUER company, and together with my colleague Stephan, we will present you the xylitol recovery and the technologies applied in this process. The recovery of xylitol is part of the mixed sugar stream within the IMPRESS project. Xylitol, a low-calorie sugar substitute, can be produced from xylose by microbial fermentation. Xylose is a pentose sugar that is the main building block of hemicellulose polymers. Hemicellulose hydrolysates are a side product of biorefineries released during the processing of nitrocellulose, for example wood. Besides xylose, also other sugars such as arabinose, glucose, mannose, galactose, and other chemical compounds such as acetic acid are constituents of hemicellulose hydrolysates. In the traditional xylitol production process, hydrogen is used to convert xylose to xylitol. In hydrogenation process, heavy metal catalysts are required as well as high-pressure hydrogen gas. To protect the catalyst, a highly purified sugar solution is required, while the sugar solution for the fermentation process does not need to be purified as long as it does not contain inhibitory substances. In the IMPRESS project investigated xylitol production process, that is shown in the block flow diagram, starts with the hemicellulose sugar solution. In the fermentation process, the xylose is converted by microorganisms to xylitol. In order to produce a crystalline food-grade product, the xylitol has to be purified and crystallized, involving a number of downstream processing steps. The process steps involve a filtration to remove biomass and organic materials from the fermentation broth. The filtered xylitol solution is treated in two separate downstream processes to prepare the solution for an SMB separation process. The output of the SMB process is a highly purified but diluted xylitol solution that can be sent to further downstream processing. Side streams are a sugar solution that is recycled to the fermentation process and a waste stream. The xylitol concentration is increased in an evaporator to a concentration that is close to the crystallization concentration. Xylitol is crystallized in a crystallizer. Often solvents are added to the crystallizer to enhance the crystallization performance. Crystals are separated and centrifuged from the mother liquor and from the solvent. Solvent and mother liquor are recycled back upstream. The solvent is recovered and returned to the crystallization process. As an alternative that is also investigated within the IMPRESS project scope for xylitol crystallization is melt crystallization. Portions of the mother liquor are added at different recycle points in the upstream process so that enriched impurities are efficiently removed from the product stream to allow production of food-grade xylitol at minimum effort and at lowered cost. The split up of mother liquor recycle to individual upstream process steps depends on the substances, in example salts, coloring substances, organic side products, etc., that are accumulated in the mother liquor. In further production steps, xylitol crystals are dried, screened, and finally packed. Crystal dust, but also oversized crystals, are collected, dissolved, and recycled upstream. A number of the critical process steps were selected from the overall production process to be investigated and developed as part of the IMPRESS project scope. These process steps are xylitol fermentation, filtration of the fermentation broth, SMB treatment of the purified fermentation broth, and crystallization. These steps are highlighted in red in the block flow diagram. The main reason is that the other steps are well-established standard operations in the food production industry. Xylitol fermentation and filtration are investigated and developed by Vogelbusch. SMB treatment of the xylitol solution is tested and designed by KNAUER. The xylitol crystallization process is investigated by Aalto and Sulzer.
In this application, selected yeast strains are used to convert hemicellulose hydrolysates with high xylose content into xylitol. The fermentation is carried out with yeast strains from Vogelbusch cell bank that were pre-selected in a previous strain selection program. A microbial fermentation for the production of xylitol has the advantage over the traditional production process in which heavy metal containing catalysts and hydrogen gas are used in the xylose-xylitol conversion process, that the feed stream does not require extensive purification to prevent deactivation of the catalysts by chemical compounds. The microbial fermentation process can cope with a large number of naturally occurring chemical compounds that are typically found in hemicellulose hydrolysates. However, in some lignocellulose degradation processes, where for example high temperature and/or sulfuric acid are used, inhibitory substances such as furfuryl and hydroxymethylfurfuryl are produced as side products. These substances are inhibitors for yeast cells under anaerobic and low oxygen fermentation processes. These substances would have to be removed in a substrate pre-treatment. Even at low concentrations, these compounds can prevent the start of microbial activity. Fortunately, in the hemicellulose degradation process that is used by our project partner, these inhibitors are generally not produced in noticeable amounts. Salts that are a side product of the hemicellulose degradation process are handled well by the used organisms. New raw materials are tested in a newly developed standard test procedure to quickly evaluate their suitability for microbial xylitol production. Here we have a short video clip from Vogelbusch laboratory showing the fermentation reactors in operation.
This video shows a typical xylitol fermentation run. Fermentation starts as a batch process that is then advanced to a fed batch process. Sugar solution is continuously added via gravimetric feeding, controlled by a feeding algorithm. pH is controlled by addition of acid and base solutions, and antifoam agent is added depending on foam levels in the reactor. Normally, two fermenters are operated in parallel with the same process conditions. Starting time of the individual fermentation is shifted by 8 to 10 hours to be able to collect samples at different stages of the fermentation during an 8-hour work day.
Another important precondition is the relative concentration of xylose within the sugar mix of the feed. Experience so far indicates that the xylose content amongst sugars needs to be at least 75% to encourage efficient xylitol production and to avoid excessive biomass production. Yeast biomass is a side product of the fermentation process that can be used as an animal feed. Due to the requirement that the xylose content in the feed should be relatively high, only hemicellulose hydrolysates from selected lignocellulose materials are suitable for the microbial xylitol production process. Suitable hemicellulose materials must have a high xylane content that ensures a high xylose concentration in the sugar solution. Initially, the fermentation process was started as a batch process. Based on process developments, the next step was to test a repeated batch process, where a part of the culture liquid is withdrawn once certain conditions, typically a high xylitol concentration, in the fermenter are reached. The fermenter is then replenished with fresh feed material and a new cycle is started. Tests have shown that a recycle of biomass by applying a membrane filtration process has additional advantages for the process. The membrane filtration is being tested and optimized as part of the xylitol pilot scale production process. In certain runs, yields of 80% based on xylose and xylitol concentration of more than 130 grams per liter were reached. A high yield is a good indicator and a must-have for an efficient fermentation process. A high xylitol concentration in the broth is a precondition for an efficient downstream process. Other factors that have an influence on the performance of the fermentation process is the production of side products such as glycerol, ethanol and erythritol. Formation of these side products should be minimized to simplify the downstream treatment process and to make the overall process more efficient. The next application is the SMB process. KNAUER received 3 differently treated fermentation samples from Vogelbusch. These samples were from the xylose to xylitol conversion. The first sample was filtered after the fermentation, the second was treated with activated carbon after filtration, and the third sample was additionally desalted by ion exchange material. Visual comparison clearly showed that the decolorization of the third sample compared to the other 2 samples. As the accumulation and thus blockage of chromatographic columns by impurities should be avoided, only the sample treated by activated carbon and ion exchange was used for further experiments. Analysis of this sample revealed a high content of xylitol and low amounts of erythritol and glycerol. The sample containing xylitol was injected on a larger column to evaluate which chromatographic separation might be applied for separation and purification of xylitol. The xylitol is eluting as last peak, which would allow, besides classical one-column batch LC, the application of simulated moving bed chromatography, short SMB. But what is SMB? The SMB technology and its application in the IMPRESS project will be explained in the following clip.
In principle, SMB is a separation technology that works using the same fundamental principles as other forms of liquid chromatography. However, the method is a little more complex. SMB systems separate substance mixtures continuously in 2 fractions. One fraction, the raffinate, is the fast eluting fraction. The other fraction, the extract, is retained for longer in the columns. In this case, xylitol is the extract. To achieve continuous separation, we add a little twist, and this is where SMB differs from conventional chromatography. We move the solid phase, which in this example is the rod, in the opposite direction to the liquid flow. Now back to the question about the fermentation broth and how SMB can be used to separate xylitol from the other components in it. In real life, it is physically impossible to have an infinitively moving solid phase. However, we can rotate the columns and thus create movement that simulates the behavior of a continuously moving solid phase. Because the movement of the solid phase is faster than the movement of xylitol through the column and slower than the movement of the other compounds present in the mixture, the columns carry xylitol in the opposite direction relative to other compounds in the mix. As the process stabilizes, there's a continuous flow of these separated components at the outlets. This process can now be operated for days, weeks, or as long as needed.
KNAUER is offering 2 different sized SMB systems. The lab SMB system can be used for proof of concept studies for later, larger purifications, but can also be used for purification of target compounds in milligram to gram scale per day. The larger pilot SMB system can be implemented into a production process of target compounds, allowing purification of target compounds in higher gram scale per day. For sugar purifications, as the xylitol purification, significant larger SMB systems are used in industrial scale. But the method development for such processes can be performed with systems shown here. Within the IMPRESS project, the purification of xylitol will be first established on the lab SMB system and then scaled up to the pilot SMB system. After that, the substances are crystallized. Sugar alcohols are very soluble in aqueous solutions, and their solubility increases at a higher temperature, which allows various crystallization methods to be applied. The crystallization methods investigated in the present work were batch cooling crystallization with or without solvent additive, semi-batch evaporative crystallization, and semi-batch anti-solvent crystallization. Crystallization conditions affect product crystal properties. Thus, CSD varies significantly depending on the applied crystallization method,
and the specific parameters used during the crystallization process. For example, increasing the crystallization time and initial temperature leads to an increase in xylitol crystal size. Here is a video clip from Aalto University and their laboratory systems.
A systematic study of the effect of crystallization parameters was accomplished in an EasyMax 402 stirred reactor station by Linden. The station has two reactors with different volumes, 400 milliliters and 100 milliliters. Temperature sensors immersed in the reactor help to maintain an accurate temperature, especially important in cooling crystallization. Cooling crystallization can be done in both reactors. In a larger reactor, evaporative crystallization can be carried out using a condenser vacuum pumping system connected to it. In addition, a peristaltic pump connected to a crystallization system allowed a continuous supply of an antisolvent required for antisolvent crystallization. Focused beam reflectance measurement is used in our crystallization studies because it allows us to obtain some kinetic data corresponding to nucleation and crystal growth rate. A highly precise chord length distribution sensitive to particle size and count is reported in real time. Next, let's take a look inside the reactor to see what is happening there.
The particle size distribution and morphology influence the product suspension filterability, drying properties, and other downstream processes. As you already know, these properties change depending on the crystallization method and parameters. That's why Aalto has 3 different filtration systems in our lab: Büchner funnel, basket centrifuge, and press vacuum filter. And finally, how to apply the obtained crystals. One of the most known applications for xylitol are in chewing gums and other dental care products, as well as in food industry as natural sweetener. However, xylitol can be used as an intermediate component for the production of multiple substances. Xylitol has applications also in cosmetic industry in cleaners, creams and lotions, as well as in pharmaceutical industry.
Thank you, Yannick and Svea. Our third presentation will introduce the route from down technology sugars into glycos and further to valuable end products. Siu-Ha, please go ahead.
Hi everyone, I'm Siu-Ha, project leader at Avantium. One of the aims within the IMPRESS project is to purify the glycol streams from whey technology. These purified glycols will be converted into valuable end products. Let's take a brief look into Avantium whey technology. Whey technology is a highly efficient one-step process that converts sugars into plant-based glycols. The monoethylene glycol, MEG, and monopropylene glycol, MPG. MEG and MPG are both important chemical building blocks for many products used in daily life. MEG is used in PET and PF resins, for example, in plastic bottles and packaging, and MPG is used for the production of functional fluids and unsaturated polyester resins, UPR. And they are also used in food, feed, pharmaceutical applications. Through our whey plant MEG, Avantium has developed an environmentally friendly plant-based alternative to this otherwise fossil-based component. This innovation also holds strong commercial potential. Our whey technology can deliver whey plant MEG, which is competitive with fossil-based MEG, both in terms of quality and cost, while showing sustainability benefits by lowering CO2 emissions and contributing to a circular economy. Avantium is scaling up whey technology, and our demonstration plant in Delfzijl the Netherlands successfully completed startup and commissioning in 2020. Now that you have been introduced to our rate technology, I will leave you with this short tour of our rate technology demonstration plant to see where it all comes to life. Thank you.
Imagine a cost-competitive solution to making plastics and textiles markets more sustainable. We are here at Ray Technology Demonstration Plant in Chemiepark Delfzijl, where Avantium turns industrial sugars, such as this, into plant-based monoethylene glycol, which is a chemical building block for making polyesters in textiles, like this, and plastics in various applications, for instance, in toys. Want to know how? We are meeting plant manager Johan Warta, who will show us around and explain the process. So what's in here?
Here we dose our sugars to reactor 1. We're now at reactor 1. That's the most important process step in our process. Here we convert sugars into monoethylene glycol.
Why is this so unique?
This is so unique because we developed a one-step process instead of a four-step process, and it's sustainable because it's bio-based. Here we have our collection vessels of the pilot plant, and that's the feed vessel to the distillation tower, where we purify the MEG. The raw materials from the pilot plant, they come here in the distillation tower, and we distillate the purified MEG on top of this tower, and that will be collected in vessels in the pilot plant again. We're at the highest point of Avantium pilot plant in this bad weather. This is where the distillation product is formed. We're at the end of the process where our MEG is collected in these vessels, and that will be transported to other stakeholders for further development in the chain.
So what is the capacity of this plant?
This plant has a capacity of 10 tons sugars a year.
So this is a real flywheel for commercial development.
Yes, actually we test here the technology, and when it's good testing we can develop the flagship in proper way.
Johan, thank you for showing us around.
You're welcome. I hope I've given you a good impression of the pilot plant where we convert sugars into bio-based MEG, and actually we can convert that into these textiles and plastics.
Thank you all for joining the new high-value plant-based products from Impress webinar. This was, in fact, the second part of a series of 4 webinars. In the first one, we discussed the challenges that Impress Consortium is devoted to solve within the 4 years of the project and our objectives. This recording will be found on our homepages. Our last 2 webinars will focus on evaluating the environmental and techno-economical sides of the concept. In the third webinar, Svea will introduce life cycle assessment process and present the Impress concept LCA. PDC will wrap up the webinar series by walking us through the conceptual process design using their expert system called ProSyn and sharing results from the techno-economic assessment of the Impress concept. Stay up to date via the Impress website and social media channels, and see you next time.